All right, this video is going to be contraction of skeletal muscles, part one. We're going to focus on two parts here: how we stimulate the muscle to contract through the neuromuscular junction, and then how they actually contract at the level of the sarcomere. So, stimulation contraction of muscle cells. To contract, skeletal muscle cells must be stimulated by nerve impulses. They are voluntary muscles, and so we send a nerve signal, a nerve impulse to the muscle cells. One neuron and all of the skeletal muscles it stimulates, because one single motor neuron can stimulate a few muscle cells or hundreds of them, depending on that muscle and the work that it does. One neuron and all of its skeletal muscles it makes up basically one whole motor unit. Also notice that we use this word neuromuscular a lot, neuromuscular junctions, for example. You're hearing the word neuro and muscular, seeing that joining of the nervous system and the muscular system here. Neuromuscular junctions specifically will be where the axon terminals join with the sarcomere, so the ends of our nerves joining with the actual muscles themselves. By the way, the specific neurotransmitter that stimulates our skeletal muscles is going to be acetylcholine. Oh, back a bit. This is just another version of this picture here, where we have two different motor units. And here we actually have a slide showing the nerve cells branching and the axons attaching to muscle fibers themselves. So although the nerve endings at muscle cells membranes are close, they look really close, they look like they're attached, they actually never touch. There's actually this space that exists between the neuron and the muscle cell, and they don't actually touch. The space in between them is called the synaptic cleft, and it is filled with fluid. Okay, so what happens at the neuromuscular junction? So a nerve impulse or action potential reaches the axon terminal of the motor neuron. So the nerve impulse gets to the end of the nerve, right? So it reaches the terminal here. There are calcium channels in the membrane of the neuron. They open and calcium enters the axon terminal from the sarcoplasmic reticulum, right? Remember we said the sarcoplasmic reticulum is surrounding our muscle fibers. Calcium enters and it causes some synaptic vesicles to release their contents. So at the end here we have these vesicles, remember those like shipment containers that are holding acetylcholine, which is a neurotransmitter. And we don't want them to like trickle out through like small channels one at a time. We want them to release the signal all at once. So we release that via exocytosis. Remember that bulk transport leading out of the cell. So that's going to release their contents. So we via exocytosis. So again, a signal came that caused calcium to enter, and that calcium causes some of our vesicles to release acetylcholine into the synaptic cleft. So now we have acetylcholine in the synaptic cleft, and it diffuses across the synaptic cleft, going from the neuron to the myofibril, and it will attach to receptors located on the cells here. Okay, so we're attaching two receptors and bind. So here we have a channel, a protein channel, and notice these green balls here, these green circles, are acetylcholine binding to the channel. Okay. So if enough acetylcholine is released, so if we have enough acetylcholine in the synaptic cleft that they bind to enough channels, the channels become permeable to sodium ions, and they rush in. So here we have sodium rushing into the muscle cell. and potassium ions diffuse out of the cell. So we have sodium coming in, potassium running out, and this is due to the channel opening because acetylcholine has attached. So again, acetylcholine binds, channels open, allowing the simultaneous of passage sodium in and potassium out. More sodium ions enter than potassium leaves, producing a local charge, a depolarization. So we're becoming more positive, less negative of the electrical conditions. And that leads to the action potential occurring in the muscle cell itself. Okay, so again, we have more positive coming in than we have leaving, even though we're only dealing with positive ions. It's sort of an interesting aspect here. And so the interior has an excess of positive ions, which reverses the electrical conditions that it used to have. This is called depolarization. And it actually opens more channels, allowing more sodium to enter. So we have lots of sodium entering the cell now, okay? It's creating this electrical current that is called an action potential, and once it has begun, it is unstoppable. It travels over the entire surface of the cell, conducting an electrical impulse from one end to the other, and the result ends up being a contraction of the whole muscle cell altogether. And while this, I didn't mean to do that. While this is occurring, acetylcholine, which originally began the process, right, because it was released from our neurotransmitter, it began this process by opening these channels. It is then broken down into acetic acid and choline by a specific enzyme called acetylcholinesterase that's present on the outside of the cells here. So we have this acetylcholinesterase enzyme that breaks down our acetylcholine, that then detaches it from, it's degraded, it can no longer attach to its receptor, and therefore the channels are closed. So we have this initial burst, and then it decreases. So we're not stimulating our muscle perpetually, we've stimulated it once, and then that stimulation is over. Okay. In order to return to resting potential after all of this has happened, we have more diffusion of potassium, and we operate a sodium potassium pump that uses actually active transport. So we use ATP to move sodium and potassium back to their initial positions after that initial contraction, after that initial imbalance. In our PowerPoint, there is a video of this working that I will not show here.